So this is a paper we recently submitted for publication, and I want to share the results with you. First of all, I want to refresh some concepts for those who are not in the field of chromosomes. So we deal with cytogenetics. Cytogenetics is the part of genetics that deals with the study of the structure, function, and behavior of chromosomes. So we have classical cytogenetics when we use conventional staining, and also molecular cytogenetics when we use molecular tools as in situ hybridization. So cytogenetics provides a valuable and irreplaceable source of information to address taxonomic, evolutionary, and applied problems. Combined with morphology, biogeography, and molecular markers, cytogenetic traits can help identifying instances of hybridization and chromosome rearrangement involved in speciation. So our subjects are chromosomes. Chromosomes are discrete hereditary units of mutation. They are identifiable, present in defined numbers with precise shapes, and they have measurable and relatively constant traits. Meiosis regulates their variability in such a way that they are species-specific. Meiotic mating provides elements for the study of chromosome homologies, chromosome rearrangement, and reproductive barriers. So here we have a diagram of a typical chromosome where the main traits we study. This is a replicated condensed metaphase eukaryotic chromosome. And here the main traits are 1, the chromatids, 2, the primary constriction or centromere, 3, the short arm of the chromatid, and 4, the long arm. So to study the chromosomes, we need to classify them into categories. There are different classification systems, but the most popular is this of Levan. To classify the chromosomes, we divide the long arm by the short arm, and with the results, we put the chromosomes into 4 categories. Metacentric or M chromosomes, where both arms are almost equal. Submetacentric or SM, subtelocentric or ST, telocentric or acrocentric chromosomes or T, and also we have chromosomes with secondary constrictions or satellites. So karyotype is the chromosome pattern of an organism expressed through a code established by convention which describe its morphological characteristics. Its graphic representation is what we call an ideogram. Here we have as an example ideograms of alloy. You know, Aloaceae family is characterized by bimodal karyotypes, meaning that in the same chromosome complement, we have 2 sets of different chromosomes, large chromosome and small chromosomes. So patterns and mechanisms of karyotype evolution may explain diversification and speciation processes through the comparison of chromosomes from different taxa. Major karyotype changes establish immediate post-zygotic crossing barriers and are expected to be congruent with clade differentiation. Our lab has been studying chromosome in the Solanaceae for a long time. This is a pioneer work from the 1961 in the genus Sclerophylax with classical techniques. We studied, many of us studied chromosomes in the Solanaceae family. This is an example of Larnax and Deprea. Here they employed just classical staining. It's just describing chromosomes. But then we started to relate the chromosome results with the phylogeny. This is a paper on Jaborosa, another genus of Jaborosa, of Solanaceae. We just overlap the chromosomes onto the available phylogeny. More recently, we started to apply stochastic mapping and ancestral state reconstruction to have accurate calculations with statistical support of the changes occur during the course of evolution. So we have the current states of the extant species, but we also can reconstruct the character state in the different nodes of the phylogeny. So we are studying right now the Physalideae tribe. This is the background. We started first studying the Iochrominae subtribe, and we found that chromosomes in the Iochrominae are very homogeneous with symmetrical karyotypes. But we know for previous work, these are the pioneer works of Margaret Menzel in the 1950, and these are drawings with camera lucida, and here we can see that chromosomes are not so symmetrical. So we wanted to compare Iochrominae to the rest of the tribe. As we know, Solanaceae shows a diploid series from X7 to X14, but the original base number for the family is a matter to be clarified. Olmsted suggested that X12 is apomorphic. This is the most frequent base number and characterizes an entire clade, the so-called X-clade, in which Physalidae is placed. The tribe Physalidae has the highest generic diversity in Solanaceae, comprising around 30 genera and more than 200 species, mostly with the basic number X12. Within the Physalidae, we have the subtribe Physalidae, which exhibits an apparent similarity in the morphology of the flowers and fruits, which makes it difficult to define their genera. Here we have 3 genera, Chamaesaracha, Quincula, and Physalideae. But in its current circumscription, the subtribe includes 13 genera. These 3 genera are recovered as monophyletic in all the phylogenies. So we call them the PCQ clade, Physalideae, Chamaesaracha, and Quincula. Of course, the most important genus within Physalidae is Physalideae, with around 90 species, including some species that are economically valuable because they are crops like the ground cherry and the Uchuva or tomatillo, the golden berry. So we have highlighted how chromosomes are important. However, most of the cytogenetic information available for Physalidae is restricted to reports of chromosome numbers and meiotic studies. So the aims of our work were to infer evolutionary patterns of chromosome characters onto a molecular phylogeny of the Physalidae, accounting for an increase of karyological characterization of the genera within this subtribe, especially Physalideae. Using comparative statistical methods, we addressed the following questions. Is there an increase of karyotype asymmetry in Physalidae? Are chromosome patterns informative enough to distinguish clades? And how many independent changes in diploid level have occurred in the group? To do so, we compile karyotype information for 42 species, including 18 new reports. We perform classical cytogenetic techniques, obtaining chromosome number, ploidy level, karyotype formula, average chromosome size, and haplokaryotype length. We also calculated the arm ratio I explained before, and in addition, we calculated also another two asymmetry indexes, the Intrachromosomal Index, or A1, which accounts for differences between arms of a single chromosome, and Interchromosomal Index A2, which explains size differences among chromosomes of the same complement. This is a summary of the studied species. In yellow, we have the Physalideae species, in gray, the Chamaesaracha species, and in other colors, the rest of the species in the tribe. So we found in Physalideae a karyotype formula with 1 to 8 M chromosomes, or metacentric chromosomes, 5 in average, 1 to 9 SM chromosomes, 4 in average, 1 to 5 ST chromosome, 2 in average, and we found telocentric chromosomes in 2 species. So we can say that in average, the formula for Physalideae is compounded by 5 metacentric chromosomes, 4 SM, 2 ST, and 1 telocentric chromosome. These are pictures of metaphasic chromosomes just to show you how they look. These are the diploid species with 2N24. This is the Aneuploidy Quincula with 2N44. And these are the Polyploid species, meaning, tetraploid species with 2N48 in Physalideae, and we found also polyploids in Chamaesaracha with 2N48. So comparative phylogenetic methods were applied onto a molecular phylogeny using the ChromoVol program. On the analyzed species, the high portion are deep and the rest are polyploids. Diploid ancestor was reconstructed for the subtribe, and 5 to 6 polyploidy events were estimated, plus 1 aneuploidy event in the case of the monotypic genus Quincula. This is the phylogeny of the subtribe with the ancestral state reconstruction for chromosome number. The black circles are species with N12, red species with N24, and green species with 22 chromosomes. Gray is uncertainties. So this is the ancestor for all the subtribe with N12. These are 6 independent events of polyploidy in Physalideae and in Chamaesaracha and Physalideae. And this is the aneuploidy event in Quincula. So aneuploidy is a chromosome alteration, a numerical alteration. When we have a different number, we have lacking chromosome pairs or extra chromosome pairs. In the case of a Quincula, we have aneuploidy because we have 2N44 instead of the expected 48. The Robertsonian translocation is the most likely explanation for this situation. In this scheme, 2 telocentric chromosomes, like this, exchange their arms, giving as a result a big metacentric chromosome and a tiny one formed by 2 short arms, which is subsequently lost. This small is subsequently lost. But an alternative explanation would be that the fusion of 2 telocentrics by their short arms with a later inactivation of 1 centromere. Both explanations are plausible, considering that the presence of ST chromosomes is one of the standing features of the Physalideae. Well, so most species in Physalideae have X12. X12 is constant in the subtribe, but we found independent events of polyploidy that can be related to processes of speciation and colonization of new habitats. Polyploids frequently have a wider geographical range compared to their diploid parents, because they have a diversity of alleles that can confer more chances to colonize new ecological niches. diversification of Physalidinae took place mainly in the Mexican transition zone here, 6.5 million years ago, when the formation of mountain ranges generated an altitudinal and climatic changes that led to the radiation of taxa. But the ancestral distribution for Chamaesaracha and Leucophysalidae, which are polyploids, is the Nearctic, and it is characterized by their adaptation to xeric conditions and present more recent divergence rates, like 5.8 to 1.6 million years ago, that coincide with the aridification of the North America during the Pliocene. But we also have polyploids in Physalidae. Physalidae angulata and Physalidae pubescens, which grow from the USA to Argentina and are also exotic in the Old World, and Physalidae peruviana, which is, of course, American, but is also exotic in the Old World. But in this case, this polyploidy may be related to anthropic dispersion. As polyploids often exhibit increased vigor, humans induced polyploidy or selected natural polyploids in order to obtain improved cultivars. Here we have these phenograms, which are the representation of the phylogenetic relationships in the phenotypic space. Here we have the A1 index, which has a strong phylogenetic signal. Intricate branches, meaning convergence. So we have in the PCQ clade a strong phylogenetic signal which separates the PCQ clade from the rest of the Physalidae, from Caliphysalis, Alkekenji, and also from the Iochrominae. In a lesser extent, we also have a phylogenetic signal in the A2 index, and also in the arm ratio a little less, separating the Physalidae from the Iochrominae, but we don't found any phylogenetic signal in the C, meaning the average chromosome length. So we cannot separate Iochrominae from Physalidae according to the chromosome size. Here we have the ancestral state reconstruction for the subtelocentric chromosomes. We do the same with the SM chromosome and the T chromosome, but I am just showing just the results for the ST chromosomes. So the black circle is absence of ST chromosomes. The red is a single karyotype formula with a single pair of ST chromosomes, and in green, 2 or more ST chromosomes. Sorry. And according to our reconstructions, the most probable state for the ancestor of all Physalidae and Physalidae had a formula with non-ST chromosomes here. But the probability of having a karyotype with at least one ST chromosome becomes higher with the common ancestor for all the Physalidae here. And this trend increases in some groups of Physalidae which present formula with more than 2 ST chromosomes here. So the karyotype changes in Physalidae would have a directional towards an asymmetric karyotype in the PCQ clade. So we have the whole phylogeny for Physalidae, and here we have the Physalidae, and within the Physalidae we have the PCQ clade, which is characterized by asymmetrical karyotype formula. Compared to the sister clades, for instance, Aureliana has symmetrical karyotypes, Diprea have symmetrical karyotypes, and Iochrominae have even more symmetrical karyotypes. So the most parsimonious explanation for this situation is that large chromosome rearrangement would have occurred when the common ancestor for the PCQ separated from the ancestor that gave rise to the clade formed by Calophysalis, Alkekenji, and Oryctis, this latter retaining symmetrical karyotypes. This is another argument that supports the segregation of these three genera outside of Physalis where they were previously assigned. So our results have taxonomic implications. Our results gave support to differentiate the genus Quincula from Physalis and Chamaesaracha. Also for the maintenance of Physalis Solanaceae within Physalis, instead of placing it in a monotypic genus as Margaranthus solanaceus. Also, the segregation of Calophysalis and Alkekenji out of Physalis on the basis of their symmetrical karyotypes, morphological features, and phylogenetic placement. And finally, support to distinguish Diprea from Physalis. As you know, some species of Diprea have inflated fruiting calyces, which has confused them to be mistaken by Physalis, but both genera can be easily distinguished by their karyotypes, as well as the phylogenetic distance and floral traits. So as conclusions, the main evolutionary trends with respect to the ancestor were an increase of asymmetry in the lineage that originated the PCQ clade, and a decrease of asymmetry in Iochrominae and Witheringia. Both asymmetry indices allow to differentiate genera within the subtribe. Overall, our studies suggest that Physalideae diversification has been accompanied by the karyotype changes which can be applied in the delimitation of genera within the group. However, to complete the chromosome evolution picture within Physalidae and confirm that asymmetry is a characteristic of only the PCQ clade, more karyotypes studies are needed in the remaining clades of the subtribe, especially in the Asian, like Physaliastrum. Chromosome number and morphology are still unknown for Melicia, Quatresia, Brachistus, and also for the genera recently segregated from Physalis, Celtalia, Archiphysalis, and Darcianthus. Our results also gave rise for questions for future prospects. For example, is there a relationship among karyotype formula and different macromorphological and ecological features as habit, niche, or life form? Because such relationships are usually hypothesized but not tested. For example, Menzel in the 1950 proposed a correlation among ploidy seed size and geographic range, suggesting that individuals with larger seeds would have higher ploidy, but this matter remains inconclusive. Another question is concerning traits of agronomic value. Do the polyploid Physalidae outmatch in size, vigor, or even environmental resilience their diploid parents? As you know, the most important crops worldwide are polyploids, so it would be useful to address this question for the ongoing improvement programs on ground cherries and golden berries. And this is so. Thank you very much for your attention.
Awesome. Thank you so much, Franco. We can open up now for questions.
Yeah.
Franco, can I ask one? Who are the diploid parents of the polyploid? Do you know that they're alloploids, or do you, yeah?
Well, that's an interesting question. I think we didn't perform any analysis to answer that question, but I think the most probable is because the most frequent in nature are the autopolyploids.
Yeah, so they're not like, it's not like Nicotiana then. Because in Nicotiana, the most frequent polyploids are allopolyploids.
Yeah, but Nicotiana are homoploids, polyploids.
No? What do you mean? No, they're allopolyploids.
Yeah, but in the case of Nicotiana glauca, it's a homoploid.
No. Nicotiana glauca is not a polyploid. Nicotiana glauca is a polyploid.
Well, there are papers that say so.
No, well, it isn't.
Probably there are different populations, because sometimes there are populations that are diploid.
We've done like 5 populations.
Oh,
they're all
diploid.
yeah, they're all
You know, the formation of
Tabacum is allopolyploid.
Sorry.
Tabacum is an allopolyploid.
The formation of allopolyploids has 2 steps. In the first step, the 2 species cross, and the resulting F1 has the same chromosome number. And in the second step, there is a chromosome doubling in order for the F2 to be oh, sorry.
That sometimes is what happens, but not always. But anyway, I just wanted to know if they were autopolyploids or allo, but you don't know.
No, we don't know.
Yeah.
More questions?
I have a question for you, Franco. This is Stacy.
Hi.
So, hi. That was really fun, and thanks also for giving a little background in cytogenetics. I always can use a refresher. I was wondering if there are particular species that you're thinking about following up doing fish on, to see if you can figure out sort of what pieces of the chromosome moved around. I'm really curious about the move from being symmetrical in the ancestor to being asymmetrical in Physalis, and I'm wondering like what chunks moved around, or if it was more of an expansion situation. So anyway, I'm wondering if you're planning to do that.
Yeah, well, small chromosomes have the tendency to be lost, and the short arms tend to be because, you know, the longest the chromosome, the higher the probability of have chiasmata. Yeah, so when you have tiny chromosomes, there is no recombination, so that is junk for the genome, and that's because they are lost. I don't know if I'm answering your question.
Yeah, sort of. It's related to that. I'm just wondering, you know, like you did with the nice Iochrominae studies, you had all that fish where you could look and see sort of where there were expansions, you know, where we had, I think there were a bunch of, maybe Rocio remembers, like there's a bunch of ribosomal sites or something that multiplied and made Sriracha have really big, chunky chromosomes for some of them. And I'm wondering, you know, if you're going to do something similar with Physalideae to figure out if some of that asymmetry came from particular, you know, expansions or moving around. You know, I don't do fish, so I don't actually know how much fish can tell you. But that's my. I don't know if I made my question more clear. Maybe not.
Yeah, I'm starting some fish in Physalidinae. So, but it's just preliminary results. So I can answer the question yet, but always, I mean, frequently the fish signals are pointing out repetitive DNA, like retrotransposons. It's DNA material that doesn't transcribe into proteins.
But I thought like it kind of gave or could give like a fingerprint and help you figure out sort of which chunks of the chromosome expanded and moved or possibly moved around.
Yeah.
Oh, okay. Oh, I think you're talking about Gish, Stacy. which is the stuff we did in Nicotiana, where you can map the different genomes in the allotetraploids. That's
Yeah,
good.
you're right. I get my fish.
It's the genomic version versus just the fluorescence version.
Yeah, it depends on the probe you use to make the fish. We usually use the rDNA probes, which are widespread in all the plant families, are not very specific. But you can use specific probes for specific genes that you.
Genomes. We use it for genomes. We use it for genomes.
Yes, for genomes, or even it would be interesting if we can do a probe for the centromeres. I think we have something like that in the lab. So yeah, to have a look at how are distributed the centromeres. If there is some signal for the quincula that it has this X11, so to know what happened with that chromosome that was lost.
Yeah, so I know that Mark Chase has just started a really, really interesting project in Nicotiana in Australia, which are all tetraploid. They're all allotetraploid, but they have variously lost chromosomes through evolution. So you get things that are down to 2n equals 16, I think is the lowest. No, not 2n, n equals 16. And he's looking at selective sweeps. So one of the things is, the mechanics of how that happens, but actually even more interesting is why it happens and what genes are maybe moved around or what genes are lost with respect to the loss of chromosome numbers. Because if you're losing bits of your genome, I mean, I have a friend who works on epigenetics and she maintains that no area of the genome is junk, that it all has a use, and I believe her. And so in a way, finding out losing something actually implies that it's not useful anymore, or that there's been some perturbation that means that you've lost something. So finding out why it happens would be really interesting. So quincula would be really good to work on in huge detail, because that's a super interesting, and no diversification. I mean, there it is, quincula, just on its little own self.
Yeah.
It's lonely. Lonely as a cloud.
Yeah.
Yeah, we also have like series of several populations of quincula, so that would be interesting.
It'd be super interesting.
Yeah.
You could figure it out.
Sandy, this is sort of related to what you said, and so there should be a Physalis genome coming out before too long, and I think Joyce knows more about this. How easy then, like if they could do GISH on it, would it be to relate sort of those cytogenetic changes to genomic changes? I mean, do people do that? I don't know, who knows, but I guess I haven't seen. I don't know how that works. It's such a macro molecular scale down to a fine scale genetic change.
Yeah, well, there is a big gap between cytogenetics and genomics. Cytogenetics is useful to describe and answer just primary questions, but more complicated questions are nowadays using genomics to answer these more complicated questions.
I guess the kind of thing oh, sorry, go ahead.
No, I was just going to say that Joyce is there. So I was going to say you should comment, Joyce, because when we did the tomato genome, you guys were doing FISH and GISH on all those chromosomes, and to do the mapping, to do the kind of gene assignment and everything. So that really is a junction between cytogenetics and genomics.
Yeah.
Right. I was going to comment because I was thinking the same as Stacy, having to be really cool to have that link with the genomics and the GISH and what came from where. And the FISH was really valuable for the tomato genome. So the Grassea genome, we're just waiting for annotation. Excuse me. And the Peruviana was going to be started before the whole COVID thing started. But it's planning to do one of the Peruvianas next.
So Joyce, do you think Peruviana is an autotetraploid?
That's what I see in the literature. that it is. But you know, the literature is confusing, and even for what's tetraploid, what's diploid. We haven't looked at whether it's auto or allo, but just based on what we've seen in the literature, it's auto, listed as auto.
Yeah. Well, from the genome, you ought to be able to tell that more or less.
Right, exactly. Eventually, yeah, right, exactly.
Here's another question. I have another question for you. Is the dates of Leucophistulus and Chamaesaracha, which correspond, the current dates of diversification of those two genera, correspond with iridification? What happens when Rocio discovers that the Solanaceae are millions of years older? Is there anything that you can...
Okay, all that's gonna change, of course.
Maybe not. We'll see.
Yeah.
I'm using the text from the Zamora Tavares.
For Pilar's here.
Yeah.
Yeah, I know, but yes, it could be like if it's if it was earlier, the diversification of that group, probably like that earlification was more later than we thought. So probably it wasn't affected by that, but it was affected by the other processes in North America.
Mm-hmm.
But it's true that a lot of the dates that we kind of cite for iridification of things and things happening end up being changed.
Yeah.
Because look at what happened with the Panama Isthmus. Everyone used to think that the Panama Isthmus closed 2 million years ago, but now it's very clear that it closed about 13 million years ago, which is a big difference.
So everything is changing.
Everything changes all the time, which makes it fun.
So this is more of a basic question, just because I don't think that much about this, is how does like asymmetry really occur? Like, I don't know, it's like, do we know that it didn't go through a polyploidization and then collapse again, kind of like go through a re-diploidization? Like, how do you actually sort those two out? And then is there like a reason why to have longer arms of the chromosomes? Is that to encourage recombination? And would that help diversification? Is that kind of the thought behind it?
Yeah, well, once the chromosome rearrangement occur, it's difficult to go back. But always the trend is to have symmetrical karyotypes.
Why is it beneficial to be all symmetrical? Is it just so things don't happen in mitosis and meiosis kind of thing, like to keep it regulated?
Well, this is all theoretical, but for example, in the Y chromosome of humans, the Y chromosome have a short arm where no recombination occurs because the arm is so short that chiasma can occur in the short arm. That is an advantage of having an asymmetrical chromosome. I don't know if I am answering your question.
Do you think that leads to diversification? Does like the asymmetry, is that like a theory that is out there?
Yeah, it's theoretical.
Okay.
Franco, I was also wondering about crossing studies. I mean, I know that Chelsea's doing lots of crosses in Physalis, but I don't think actually we found, I can't remember, Chelsea probably remembers, if we found that many like classic biosystematic studies. in Physalis or even Quincula or Leucophysalis or any of these things? Are there many out there, or is this sort of a big gap? Because then I'm thinking, you know, maybe we could actually use your cytogenetic survey along with the phylogeny to sort of make better predictions about where things might cross or not cross.
Well, according to the theory, species with similar karyotypes are able to cross. But this is just theoretical. We have to test these theories.
Well, actually, all the Iochrominae, I think Stacy can tell probably that the Iochrominae that have all very symmetrical chromosomes are very easy to cross, even between different genera, right?
Yeah.
Yeah, so I was thinking, I was wondering if then it probably isn't that way in Physalideae, where it's more variable. Although I will say that, you know, having seen the results that you all got for Iochrominae, there are, we didn't cross the weirdest ones. Like Sriracha has much bigger and weirder chromosomes than the other ones, if I remember correctly. And that one's, you know, big and woody and hard to grow, and we haven't crossed that one. So we crossed things that are definitely related, but we didn't structure the crossing based on karyotypes, basically. We didn't try to cross the weirdest karyotypes. I just crossed what I could grow in the greenhouse. And so Vasobia and Iochroma cross, and they're very distantly related within the subtribe. But I think they both have kind of similar symmetrical chromosomes. And of course, they're all 2n equals 24.
Having a different chromosome number is the strongest barrier, because when you have two parents with different chromosome numbers, the homologous chromosomes cannot pair during the second stage of meiosis.
But so symmetry. So thinking about symmetry in that way and diversification. If you look at Solanum, so Solanum, I couldn't remember, but Rocio reminded me that Solanum has very symmetrical karyotypes. And some of those you can cross easily, like within clades, but other ones which have equally symmetrical karyotypes you can't cross. So you can't cross a tomato with an eggplant without fantastic difficulty and perhaps tissue culture. But so it's kind of there's more to it than that.
In that case, it's always chromosome doubling is involved.
No, I'm not talking about wanting to do it. I'm talking about the possibility that you would. I'm sort of trying to go back to what Chelsea was asking about whether chromosome asymmetry increases diversification, and that's probably, I think that the jury is probably out on that, because if you look at who's diverse in Solanaceae, it's Solanum. And they have symmetrical karyotypes.
Yeah, you're right. More questions to be answered.
Yeah, I know, it's too many. Well, you're there, Franco, and you can do all this stuff so beautifully. It's great.
I hope so. I just want to go out.
So does everybody. I think Quincula. I think you need to work on Quincula. I think that would be a really cool one. And then a genome of Quincula, Joyce. How about that?
Sure. Add it to the list.
Up for it.
We can send you seeds.
I have another question, Franco. So you've probably dove into Margaret Menzel's paper a little bit more. Do you think it follows because she did a bunch of crossing and a bunch of different karyotypes? Does that hypothesis of they have different karyotypes so they don't cross, does that hold true within that paper? I can't remember. Or did she actually address that directly?
I tried to track the vouchers specimens she used, but I couldn't found it. So I'm not 100% sure of the identities of the species she used for the crosses.
So didn't she just have some SP and a number for some of her crosses? She didn't give a name to some of them.
And after her works, there were a lot of name changes. So I don't know. I contacted the curator of the herbarium, but he didn't found the voucher specimens of Menzel's crossings.
Is there any more questions? Maybe basic questions about karyotypes too are also welcomed. All right, well, thank you, Franco. Next week is Pilar. And
2 weeks.
2 weeks. Oh, I always do this every week. Nice. And she'll talk about some domestication and cultivation work that they have been doing in Mexico specifically.
Great talk, Franco.
Yeah, thank you, Franco.
Thank you.
Franco.
Thank you for your interest. Thank you for your attention.
I look forward to getting the paper to review.
Okay, bye. See you in 2 weeks.
Bye.